Welcome to A Fork in Time, the alternate history podcast.
Welcome back to A Fork in Time, the alternate history podcast. Don sitting in the host chair today and very happy to be joined once again by Eric Rush. I guess technically that's Dr. Eric Rush, so don't want to forget that you have an honorific there in front of that, Eric. Thanks for joining us again, Eric. I know that we enjoyed doing the episode we did before, talking about the polio vaccine, the Salk vaccine, and other derivatives, and I've been excited to have you back to talk again about another topic you suggested. So That's where we're going today. So since it was your topical suggestion, I'm going to let you sort of explain what it is we're going to be talking about. We'll take the break and then we'll come back and actually dive into what we do here on A Fork on Time, which is looking at what happened and then what didn't happen or could have happened.
Great. Thanks, Don. And it's great to be back. I had a wonderful time with the prior episode and happy to be back for a second round. So today we're going to be talking about what if the Human Genome Project never happened. I think those of you who listened to the previous podcast will recall that my day job is as a clinical geneticist. And so genetics technology and the products of the Human Genome Project are really near and dear to me because that's kind of what I do every day. And I think we have an idea that projects like the Human Genome Project were so obvious to everybody and so necessary that there was never any possibility that this wouldn't occur. But as it turns out, the truth is a lot more complex. History is a lot more complex than that. So we're going to dive into a specific fork on the what if after we come back from the break.
All right. Looking forward to that. I'm looking forward to going along on the ride here. I'm in the driver's seat, but I'm about to hand Eric the wheel when we come back. So hope that you stick with us after the break and we'll be back shortly.
Would groceries delivered to you in as fast as 1 hour save you a trip to the store? Instacart makes that possible thanks to personal shoppers in your area who hand-select items based on your preferences from the stores you love. And shopping multiple stores is possible on a single order. Instacart picks the freshest produce and even keeps your eggs safe, all while finding everything you usually buy, providing smart suggestions for new items, and even highlighting deals to help you save money. And now you get free delivery on your first order over $35. Let Instacart know we sent you and help support our show by following the link in the show notes. Instacart. Groceries delivered in as fast as 1 hour.
Welcome back to A Fork in Time. Don leading the effort today, but I'm happy to be joined, as I mentioned in the opening, by Eric Rush, who has been on the podcast before and suggested this topic. And as Eric outlined, we're going to be talking about the what if of an alternative for the Human Genome Project. So maybe a good place to start. We tend to start with the historical what did, Eric, but we don't want to go too deep into that, but it's necessary, I think, for the what if here. So what in the world is the Human Genome Project and what actually did happen in history before we talk about what didn't happen?
Yeah, so the Human Genome Project is really biology's first foray into what we would call big science. Prior to the Human Genome Project, a lot of the big science had been more in things like physics and astronomy, but this was the first foray into big science for biology, insofar as it leveraged a lot of different research centers in several countries to provide a sequence of the human genome. The human genome being the entirety, or in this case, really the near entirety of all of the genetic information that's contained within human genes. And that at least one genome. Now, again, we're all a little different. And so there's always a little complexity there. But what this started out as is an effort from 1990 until 2003, when the project was declared completed, to create that sequence. And that it worked. It was a successful project as far as that goes. But like a lot of things, it starts with a fair bit of negotiation before that. And even though science is important in its own merits, we can never take science out of context. and we can never take science out of politics. And I think that that because of that, we have a lot of differences in sort of how we think about the pure science going as opposed to perhaps how a lot of the setup occurred. And so, this is an interesting topic of discussion, partly because it really wasn't initially a National Institutes of Health initiative. And I think that's one thing that people think is sounds really odd, because we kind of associate the Human Genome Project and the current Human Genome Institute as being strongly associated with the National Institutes of Health, and they certainly are at this point. But really, the past, the history of the Human Genome Project starts not with the National Institutes of Health, but with the Department of Energy. Which may sound very interesting.
Yeah, we were talking about that off podcast, and you had mentioned that, I think, in some of the email exchanges that we had. And it's like, okay, I didn't know that. Now I need to understand why that is, because it doesn't immediately click in your head that something related to what seems obviously like medicine and biological science ought to be from the same guys that bring you power.
Right. Exactly. And not just any power, nuclear power,
Right.
in a lot of ways is kind of where this started. But we can even go back a little further than that. And so I think we would even go back to as early as the late 70s. And the reason I would go back there is because the late 70s were kind of the first time that which we actually had the technology to do DNA sequencing. On a larger scale. You could do it before, but it was very laborious, and so a technique was invented that we now know today as Sanger sequencing, named after Frederick Sanger, and that sequencing allowed us to sequence larger stretches of DNA and do it a little more efficiently. But it was still, by today's standards, extremely expensive, extremely slow. But it's accurate. It's an accurate way to do things. And so in the late 70s, a couple of viruses had their genome sequenced. And these were very small viruses. We talk about base pairs in genetics. And so, each base pair is just one of those little letters, those A's, C's, T's, and G's. And each one of those is married to its counterpart on the other side.
Which, as I understand it, is a particular counterpart for each one of those.
It is. Everyone is matched. So the C's to the G's and the A's to the T's, everything is matched appropriately based on the chemistry. And so your first viruses that they sequenced were only about 5,000 base pairs long, and this was the first one was a virus that actually infects bacteria, or we call a bacteriophage. Those are really peculiar little viruses, and this is one in particular called Phi X174. 5,000 base pairs long, and that is really, really little, but we have to start somewhere. And just by contrast, the human genome is not 5,000 base pairs long. It's about 3 billion base pairs long. So just a little bit different complexity there.
A couple of more zeros on the back end, which make a big difference.
Right. And most of these bacteria, their genomes are on one little circular piece of genetic material. It's a little bit easier than the 23 pairs of chromosomes humans have. But then by the early 80s, there was a couple of really interesting papers that were developed that said, well, what we need is a systematic way to make these DNA, what we call linkage maps. And this is not the same thing as a Human Genome Project, not sequencing the whole genome, but creating linkage maps. And so what a linkage map basically is these markers where we link to are just these areas that have a known location within a chromosome. And you can, if you space them out in certain intervals along the chromosome, you can link them to certain genes that may cause disease. It's not the same thing as actually sequencing the gene, but you could at least know approximately where it's located, so you can do a little better research on it. And so it was hypothesized at that point, if we had higher quality gene linkage maps, then we could more efficiently do rare disease research. And that's true. I mean, that was a true thing to say, but that was really more it was around 1980 when we started hearing calls for that. And mapping increased. There were some more markers that were created, But then it really wasn't until about 1985 that there started to be calls for a Human Genome Project, or something that looks like the Human Genome Project. One that was actually done at UC Santa Cruz. There was a whole conference that was convened that will say, this is the Let's Do a Human Genome Project. And this was 1985. And everybody met, talked about it. Then NIH said, basically was like, we're not interested at this point. And because a lot of people really, and even during the Human Genome Project, a lot of people, very reputable scientists, said, we think this is kind of a boondoggle. We don't think this is really good science, and we don't think this is going to pay off. But so NIH wasn't really interested in pursuing. The next year, in 1986, there was a workshop that was organized in Santa Fe, New Mexico, by the Department of Energy, by this Office of Health Environment Research. And again, this Office of Health Environment Research had a long history of doing research on the effects of radiation and chemical mutagens on genetic material, because we know that radiation can destroy DNA. We know that certain chemical mutagens can prompt mutations, and that can cause issues for human health. But the understanding was a little limited by the fact that there's a lot we didn't know about the human genome. So there was interest within that entity of the Department of Energy for this. And so eventually what was created a few years later in 1990 was a collaboration between the Department of Energy and the National Institutes of Health. And so they co-created the Human Genome Project and co-ran it. And it was really, at least from the outside, and I'm too young and not really the right kind of geneticist to be involved in the actual Human Genome Project. But from what the outside it looked like during those first couple of years is the efforts were more or less run in parallel. And then in 1993, a new director of the NIH side of the Human Genome Project, Dr. Francis Collins, who's now he's director of the National Institutes of Health. And Dr. Collins, very distinguished scientist, long history in gene discovery. He was part of the group that discovered the gene location for cystic fibrosis. And so he has a long history of being involved in this. And he was placed as the director of the NIH portion of the Human Genome Project, but then the overall effort as well. So that really kind of put it a little more on the NIH side, and you could sort of feel it over the course of the rest of the project being just a little pulled more towards the NIH side of things. Again, from my perspective, it definitely looked like that. But over the course of the Human Genome Project, not only was the Human Genome Project mostly completed, and again I say mostly because we talk about the human genome being complete in 2003. In actuality, it wasn't totally complete in 2003. It was probably more like 93% complete. There's a good 4 to 7% of the Human Genome Project or human genome that's just not accessible to the technology at the time. Some of it's still not very accessible. But it was more or less complete. But one of the problems with it is we just didn't have the technology in the late 80s when this project was being conceived of to really do the project. It was well known during the Human Genome Project that there was going to have to be new technology developed to speed things up. And at first it was automation of the Sanger sequencing, that old school sequencing that we used during that. But then the second big breakthrough was what we now call next generation sequencing. And next generation sequencing is what I use every single day for genetic diagnoses of patients with rare genetic conditions. And so that's definitely one of the dividends. But those are the technologies that were required in order to make the Human Genome Project a possibility.
Okay. And it also just, I know we're focusing there on the U.S. history, not to go too deep into this, but there was also a global effort. This was in cooperation with other institutions around the world. So it wasn't just the U.S. going it alone on this. I know that when the actual, I guess the first publication of this, well, I think it was a joint, then President Clinton and Prime Minister Blair, for example, of the United Kingdom, were the ones who sort of said, yeah, if I recall correctly, like, here are the books. Here's it actually printed out. I think that was sort of the gimmick. There was like a bunch of A, T, C's, and G's in a book, even though obviously it was being stored in other ways as well.
Right. But you're exactly right, Don. It was an international effort. I think the U.S. provided a significant share of the funding. A huge share of the funding. But there were groups in the United Kingdom, there were groups in France, Germany, China, Japan. So there were groups outside of the U.S. who are also working on the Human Genome Project, which again is one of the reasons why we consider it sort of biology's first foray into big science.
Yeah, I read several things as I was preparing here, talking about, it was sort of it had a moonshot sort of quality to it in the sense of, we're not quite sure what we're going to get out of this, but it's a big project. It's going to require a lot of effort. We know it's going to require, and again, I think the what you mentioned there, sort of the similarity there, it's going to require technology we don't quite have yet. We're going to have to develop some technology to be able to do this. And we'll talk, I think, a little bit more as we get into the historical what if about the importance of that technology. You've already, I think, sort of foreshadowed that a bit. But then the other thing I just wanted to hit just very quickly on, again, coming on the what did, ultimately, and you were sharing this with me off podcast, it wasn't until 2013 that it was finally declared that basically this information is in the public domain. It's not something that can be commercialized directly in the sense of the raw data of who we are as a species and what the combination or sequence of those letters are, because that's naturally occurring in nature.
Right. This was always a source of a bit of tension, amongst the different stakeholders within the Human Genome Project, both public and private. Is the idea is what are the economic considerations of the Human Genome Project, and there are certainly economic considerations, but ultimately what was decided, and this was again the Supreme Court decision, and this is a matter of public record. In 2013. So this is a Supreme Court case called the Association for Molecular Pathology versus Myriad Genetics, and that's the only mention I'm going to make, because that's in the public domain as a Supreme Court decision.
Right.
But essentially, what the gist of it was that you can patent certain techniques, you can patent certain products based on genetic technology, and you can patent certain constructs that are made, but you can't actually patent the gene, the naturally occurring gene itself. Because it's naturally occurring, it's not considered intellectual property. And so, but what that does is it allows a little more flexibility for people to innovate and for people to investigate. The other thing it's done as a downstream, again, in my opinion, is it's also reduced the costs of genetic testing considerably. And this is kind of on the baseline kind of retail level, both from a research standpoint, and then when you're just seeing a patient in the genetics clinic. It's reduced the cost considerably. And so our testing is now, we don't have to hit people with, hey, this is going to be $10,000. I mean, that essentially doesn't exist anymore, which is really good.
Yeah, so obviously the medical applications there, does that also translate over into like the things that we see that are like the Ancestry and the 23andMe kind of things as well? Does that same thing play over into that space as well?
Those sort of business models are a direct result of information that's collected in the Human Genome Project, no question. And because a lot of those are based on one particular type of genetic information. These are sort of what we call mapped single nucleotide polymorphisms, or what we'll call SNPs. And
for our listeners, there will be a test later, so hopefully you took a good note on that.
Well, and what that is when we talk about a SNP, or single nucleotide polymorphism, it's just one of those little letters. It's one of those little letters. We know where it is located within the human genome, and because we say polymorphism, poly meaning many, we know there's a tendency for different letters to be found in different people. And because there's differences there, we can use it to sort of make decisions on, and some of these can be decisions on, on what pattern is more likely to be associated with, what certain ancestral group. And so those are the ways that it's used in those in those sort of contexts. Those have been definitely a direct result of that. And I think that in some cases the science is a little soft with those sorts of applications, but there's still very much an outgrowth of the Human Genome Project, and kind of not only just in the technology used, but also in the public consciousness, right?
Right.
And so I think that I will say, this is only my opinion, but I think there are others that share it. My opinion is that the that the Human Genome Project was a net benefit to humanity, and in fact, a huge benefit to humanity. But having said that, I still think in many ways we oversold it. Because I think when we first started out, and even when I was, in the 1990s, I was, mostly in high school and college, and they were talking about, we're gonna learn everything you need to know, not just about rare genetic diseases, but about your risk for heart disease and your risk for cancer and your risk for Alzheimer's disease, and in reality, we have discovered a little bit of those things, but really, the Human Genome Project has not totally explained all of the reasons why people have. We've nipped around the edges a little bit. There are certain types of really early onset Alzheimer's disease that we do know that genetics play a strong part. But for just the regular person who develops Alzheimer's disease, when they're older, there's a lot we still don't know about what genetic contribution is there. So I think we did oversell it a bit.
Yeah, okay. And I'll share a little bit towards the end, just a little bit about how those particular technologies have touched me. They've actually impacted my life. So I'll save that as a teaser for the end of the episode. Maybe folks will stick around for that if they're interested.
That's perfect.
Right. We talked about the what did, but this podcast is all about the what if. So
what if?
So where do you think is the right place to look at where there might have legitimately been a fork that would have caused us not to have what we have today?
I think the most likely fork happens before the Genome Project even starts. Again, this was a collaboration between two large governmental entities, and that success in collaboration was by no means a foregone conclusion. So I think it's very easy to imagine a history where the Department of Energy says, we're really interested in doing this. And then like they did in 1985, the National Institutes of Health say, we're not really that interested. We don't think that this is where our dollar would be best spent. And so they say, thanks, but no thanks. And so the Department of Energy says, well, we really can't do this on our own. And so they eventually table it. And no Human Genome Project starts in 1990. So what happens then? What probably happens at that point is some of the very early first steps that ended up being integrated into the Human Genome Project would have still occurred. And so I'd mentioned to you, Don, a little bit ago about these what we call linkage maps that are kind of the road maps along the way. And so it's a really basic map of kind of where certain parts of the genome are located within the individual chromosomes, so that individual scientists who are researching individual genes can more easily pinpoint the approximate location of that gene, so they can do their own individual research. I suspect that those maps would have still been created, and that we would have had progressively higher resolution maps. What was eventually created, so as a part of the Human Genome Project, the idea is that there would be maps that were created in what we call the 2 to 5 centimorgan. So centimorgan is a derived measurement that is roughly equivalent to a megabase, or 1 million base pairs. And so in this case, 2 to 5 centimorgans between those markers is roughly about 2 to 5 million base pairs. Well, that's a pretty large area of real estate when you think about it. What was eventually created as a part of the Human Genome Project by 1994 was to narrow that to about 1 centimorgan, or 1 million base pairs between those markers. Well, that's interesting, and that's a lot nicer. I suspect that would have still happened. I don't know if it would have happened by 1994, but it probably would have happened, at some time in the mid 90s. And that would have given individual researchers the opportunity to still do their own individual research. And people were still doing their own individual research during the Human Genome Project. But I think that would have still proceeded. But the actual sequencing, kind of from quote unquote start to finish, even though we know it's not a complete genome, that would have been where the stalling would have been, where we wouldn't have had that information right away. And we also probably wouldn't have had the reason to develop the higher throughput sequencers, what we call next generation sequencers. Again, I think they would have eventually been developed, but they're just the reason for them wouldn't have been developed right away. So I could easily see a history where we have these higher resolution maps. People are kind of plugging in genes kind of one by one as they start doing that research. I think there might be some companies that would come along and might say, "Hey, we want to work on this set of conditions," and they might then try to go about patenting that because the legal framework would have probably been different too.
Right.
We wouldn't have had the reason to kind of push for this being more in the public domain, and so that might have been a business model, in the say late 90s, early 2000s, to say, "Hey, we're gonna have this group of genes that are going to be kind of our genes, and this is our intellectual property, and we're going to some way monetize the downstreams from these genes." And that might have worked for a while. I think eventually the same decision that was made in 2013 would have been made, but maybe not even at this point. And I think that a lot of our current generation of sequencers, and we've gotten to the point now where we've gone from the next generation sequencers that we use kind of on a day out basis to what we're now calling third generation sequencers. Those are just starting to come online now, and have some nice kind of different aspects to them that are really adding to kind of the overall effort, and that those probably wouldn't exist at this point. I suspect that we would be at a position where we're kind of back ending our way with technology that was developed later into something that looks a little like the results of the Human Genome Project, but it would have happened a lot later. We might still be working on that at this point. And then all of the kind of the downstream effects, aside from just the sequencers themselves and the genetic information itself, a lot of things like rare disease treatments have been developed based on knowledge that have been created either through the Genome Project or kind of tangentially related to the Human Genome Project, including some of the bone diseases that I work with on a daily basis are really kind of directly dependent on knowledge that relates to the kind of underlying understanding of the gene itself. And one real success story in that recently is in cystic fibrosis. So the cystic fibrosis gene was first mapped out and started to be understood kind of late 80s, early 90s. It was kind of one of those first genes that we started to understand really well. And a lot of the treatments that have been developed in the last 10 years for that have been based on kind of that understanding of how that gene works. Including the most recently approved medication, which has been truly amazing. That really has to do kind of with ways that the compounds can restore the way the faulty gene is functioning, and so you sort of kind of partially rescue the condition. Again, that's amazing, but we can't do that kind of amazing stuff if we don't understand how the genes work.
Right. And so again, it's that, and to me, this, I think I told you this when we did the vaccine episode. I dutifully avoided biology after 7th grade. I did a lot of chemistry, did a lot of physics, but and I'm married to a physician assistant who's medically trained. So, she had plenty of biology. So I had a go-to source there for biology if I needed it. But to me, it's this struggle that exists. And I think this is a good example of that between, basic science and applied science. That's the tension that I feel all the way through this is, eventually we have the applications that come out of what we learn, but is it worth spending the money on just the basic science, not knowing if we're going to get the application? Or do you go from the other direction of somebody, like you said, an individual commercial entity saying, we're going to focus on this because we see the application, we see where the money's going to be made for this, and the science becomes tangential to the commercial need versus the science being primary and the other things flow off of that. What's primary and what's tangential sort of get flipped back and forth in this equation.
I think they do. But I think the Human Genome Project is even from people who have criticisms of it at this point, I think have to acknowledge that the basic science that's been done here has paid off in terms of human knowledge, and it's also paid off economically. I heard one statistic say that for every dollar that was invested in the Human Genome Project, $141 have been created.
Wow. That's a nice ROI.
That's a really good return on an investment, and I think that really underscores the benefits of again, this is my opinion. It underscores the benefits of government funding of basic research,
right?
And I think and I think it's no secret to anybody who's been thinking about this that one of the kind of the secrets in the sauce of kind of American innovation and a lot of our contributions in science and especially health sciences over the last 70 years were that the federal government decided, hey, we're going to fund scientists to do this sort of research. That is a good model for how things work. Is it a perfect model? No. Are there issues with some of that? Absolutely. But I think fundamentally, the National Science Foundation and the National Institutes of Health have been an extraordinarily successful way to fund science.
Yeah, I would agree with that. And again, not to go too far down the political path, because that's always a risky place to go, and we try to avoid that here. But it is relevant. Politics was involved, as you pointed out, and one of the interesting things there is that went across multiple administrations of different parties. And so this is also one of those areas where you see everyone sees the common benefit of it, because this is not a big R, big D kind of thing. This is a big H kind of thing. It's about humanity and learning about ourselves and learning how what we can in the area of medicine. And I do say this, and I'll state this is my opinion too. America is one of the most interesting things to me when it comes to medicine, in the sense that we have arguably the absolute best healthcare in the world in terms of technology and some of the things that we can do. And then we struggle on the access side. So, how do you want to measure the system by the upper end quality? American healthcare goes up against anybody in the world, if you want to use that measure. If you want to look at it in terms of access or other things, or even basic health, well, that's a different measure and probably scores out differently than we do when we look at just the high end quality.
I think you can definitely say that when it comes to innovation. I think we lead the world when it comes to healthcare innovation from a technology standpoint. And I think, as you say, where we lag is in outcomes, and part of that is certainly the access to healthcare. We could have an entire. Actually, this just gives me a totally different idea for another for another episode of this. Now that we're thinking about that, we'll talk about it later, Don.
Fair enough. All right. So again, one of the major departures, just to summarize back, I always say if I could explain it back, that normally means that I understood it. Is that your suggestion is that some of the things that we have today, it's not that they would not have come to be, they would have come to be later and probably not quite in the level, for example, the technology that you mentioned. It's going to eventually probably come along because there's a need for it, but there's not the funding that's coming inside of a major program and the desire for it to be done parallel to that, so that, three billion, I think, was the number that you threw out there. The amazing thing to me, I was doing some research, is somewhere in the neighborhood of about 100 that we're still missing, right? That's why we can't call it complete. There's like these little small segments of it that, if you will, small segments of the road that don't want to be mapped, kind of thing, or that are very difficult to map for some reason. I heard something about what biologists call the stutter, where there's like this one area where there's like the sequence is just the same thing over and over and over again there, and that creates a challenge for reasons that it does.
Right. Well, it's what we would call highly repetitive sequences.
Yeah,
is the word we use, and our current technology is not very good at being able to sort of lock on to those highly repetitive sequences. So they're a little resistant to even next generation sequencing. But third generation sequencing does a little better job with some of those regions. Yeah. And so we might still get there.
Yeah, that's sort of cool. When you think about a number like 3 billion, I mean, that's just that's a number, all the various ways that we get our heads around these large numbers, that's a big number. And that's right. That's inside, not just inside each one of us, that's inside each one of the cells inside of each one of us. I mean, that puts it even more context.
Absolutely, and if you look at this from the standpoint of cost of the human genome, you could say, well, about approximately 3 billion base pairs in the haploid human genome, and $3 billion spent on it, so about $1 per base pair is kind of what the cost would be. Now the cost per base pair is less than a penny.
Wow.
Just in terms of cost, and that's really even the cost of a megabase, of a million base pairs of sequencing, is really still less than a penny. So we're, our genomes are now, if you want to get a raw genome, a human genome is less than $1,000.
Wow. That number astounds me. I'm gonna throw in my personal story here, and then we'll come back to the other part of the what if, because this actually has a personal what if for me. I may have shared before on the podcast. I'm not certainly. This is nothing I'm ashamed of or not willing to share. I was adopted at birth, And so in the time that I was adopted, it was a closed adoption here in the state of Texas. And so one of those instances where I went until my adult life really not knowing anything about my biological heritage. And I'll say, as I always say, I was blessed by God to be given the adoptive parents that I have. My parents are my adoptive parents. No ifs, ands, buts, candy, or nuts about that. And I feel very blessed with the life that they gave me. So this is not one of those instances where I needed to find my biological family because these horrible people had raised me. It was quite the opposite. But eventually, you do become curious. And the reason that we became curious is that my daughter, when she was in utero, she's the co-host on this podcast. I'm not going to go a lot of things. There was an issue while she was in utero that caused chromosomal testing to need to take place. Right. And it was ruled out that it was not an issue that was a genetic issue, genetic defect. In fact, that was good. But, every time that they were looking at me asking now about these genetic traits, what can you tell us about this? And what can you tell us about that? I'm like, I don't know. I'm a wild card. I can't tell you anything. And so, it wasn't so much the need to try to seek out my biological parents, but to really being able to answer those questions for medical reasons for my daughter caused me to think about it slightly different than I did in the past. Well, along comes, I guess this was now 4 or 5 years ago, along comes companies like, I'll just mention them here, don't get anything for this plug, but 23andMe, where they're like, hey, find out what your, in my case, what even is my ethnic background? I don't even know, I can guess. Right. But, and so Alexis for Christmas got me the testing kit. So I send off that test and do that and come back. I guess it took about 6 to 8 weeks for that to come back, which again is part of the technology you're talking about. The fact that they can even do that, the technology exists for them to be able to do that. And as you and I talked a little bit off podcast, they focus on just elements of that. That's not mapping my whole genome, obviously.
Correct.
But relatively inexpensive process. And so I find out that which I thought the case was, I am very Caucasian, Western European. That very much is my background. I'm right. The other things that were there, but now trying to condense the long of this story. So that was cool to know. Okay, I'm a Scottish, Western European, Caucasian extract, very little of anything else. Sort of makes sense. That's just good to know. Was about 6 months later, because of the connections where now, if you opt into this, you can have your DNA compared to others there on the site, actually ended up getting connected with and discovering the person who turns out to be my biological half aunt. Oh. And as a result of that, she was able to figure out who her biological father was, who happens to be my biological grandfather. And through a lot of connection there, we were able to at least and she'd been working on, she's also adopted, she'd been working for 25 years with genetics and with other means of trying to get to this. But because of this technology and because of me testing, because if it had not been for me, because I filled in a gap for her in terms of what she had been able to identify, she was able to identify her biological father. I was able to identify my biological father. Still clueless on the maternal side of things, but that goes. But so this has had a direct impact in my life. If there had not been a Human Genome Project, I probably could not have related any parts of that story that I just told you. So this is, one of the reasons that it's one of the ways it's directly relevant to me, which is one of the reasons you didn't even know this until we're talking here right now. One of the reasons this was an interesting topic for me when you suggested it was I realized that we all have a personal connection to it. It's the human genome, but I have a very personal connection to some of the things that came out of the technology because it directly impacted my ability to ascertain my genetic descent. And in this case, a little bit of my biology and learn some of my biological ancestry to go with the ancestry that I identify with, which is my adopted ancestry. And that story is true for me. That story has to be true for countless others. So again, that's one of the what ifs here. I'm not unique in that respect. And some of that, by the way, has some nasty sort of Maury Povich, Maury show kind of thing of, and people finding out things that aren't true as well. That's the flip side of this. But it we're more informed not just about our genome as a species, but I'm informed about my genome as an individual, which has made a difference.
Absolutely, and that's so it's such a fascinating aspect of this that really personalizes it so much, and this is part of what we do when we're doing informed consent for people when they do genetic testing. So in some cases we have to tell people, listen, this will tell us about relationships between people, and will also tell us if those relationships are not present. Because we know it's not common, but we do see things like false paternity and things like that, and those are those from not necessarily medically problematic, but from a social construct, those can be really devastating to people. And so when you establish those relationships or lack those relationships, that prompts a lot of different questions. And so one of the things, so I actually have done that as well, out of kind of pure personal curiosity, and I've been contacted by a number of people who are looking for answers very much like what you were interested in, Don. And I come at from kind of the opposite of, I have a very extensive family tree that my father did, a number of years ago. And he's not so interested in the genetic aspects of this. He's a little interested, but not so interested. But my mother's interested, and my father's younger brother is also really interested. So my uncle on that side. So I kind of have on both sides. I have my mother, and then I have my dad's brother and my cousin, his daughter, that are also kind of involved there. So we've established those relationships, so no drama in our family. But when I have people that say, hey, I'm adopted, or I'm trying to learn more about my family, I see you have these last names in your history. I'm curious as to where this might be coming from. And it's very easy for me to say, well, are you related more closely to my mother, or are you related more closely to my paternal uncle? Because that'll tell you a little bit about kind of what side it's coming from. And so I've had a couple of people that I've fortunately been able to help you with a little bit more information. And so that's from my side as well. It's kind of cool to say, hey, as it turns out, we're second cousins. I don't know how yet, but we're second cousins. And so that's neat.
Yeah. And even before you use the term sitting organ, I learned what that was as a result of the way that, that's how overlaps are calculated, and certain amounts there indicate certain types or affinity levels of relationship. Like, for example,
Right.
The person that I mentioned, which turned out to be my biological half aunt, I think first identified as a first cousin. It's in the same range as the first cousin because of
Right.
the degrees of separation there. And then the other thing I'll just mention, again, this time back the personal aspect of this, I constantly now get these updates, and apparently it's that old Kevin Bacon 6 degrees of separation thing. Apparently everybody in the world is somewhere between my third and sixth cousin, kind of thing. You quickly discover, because we all share that common DNA, but we all have this overlap of the other DNA that's what's unique to our family trees. And that's an interesting part of that as well.
Well, and that's one of the that's one of the things that I do like about what we've learned about the Human Genome Project is it tells us that when, as humans we're all related to one another, and we should probably treat each other that way.
Yeah, there is that social aspect. So,
Yeah.
So what else would be different in this alternate forked timeline if the HGP hadn't gone the way that it had? Anything that of relevance that we've missed?
Yeah, I mean, I think I look at this also from sort of my day to day. My job would look a lot different today than it did. So my job today would look a lot more like the way I was originally trained. So I was originally trained as what's called a clinical dysmorphologist, and that's its own kind of interesting history. And so dysmorphology is the study of differences in the way humans develop. And so classical dysmorphology was developed in the late 60s when we didn't have a lot of this technology. And so people diagnosed patients simply on recognizing patterns on examination. If this technology didn't exist, I would be functioning much more as a classical dysmorphologist than kind of the applied molecular geneticist that I really function as today. Not to say that we don't do any classical dysmorphology today, because we do, but it's very much a different kind of a different feel. So for me, it would absolutely change the day-to-day of my job, and in some ways that would make it a lot more challenging. But in some ways that could that would actually make it in some ways make it a little easier, because we just had a conference, and one of our very, very senior colleagues made this kind of off the cuff comment to say, well, in the past I was never wrong, because there was really no way to confirm whether a person was right or wrong, because the technology didn't exist yet. And so you said, I think it's this, and everybody said, okay, sounds good, that's what it is. And you didn't really have a way to check that. And right. So, it's in some ways it's nice. In some ways it's nice to have a check against you, but in some ways it's also nice to well, I guess I'm never wrong here.
And you can't prove me wrong. Right?
Yeah. Exactly.
So, I'll ask once again if there's anything else, but I also want to ask one other question that sort of relates to this, but relates to other things. So I'll ask the other question first. Anything else that we missed here that you would want to make sure we included in the forked
I think we've
option?
got it's a pretty good deep dive into the history and the kind of the what ifs of the Human Genome Project. There are some other possible departures here, but I think starting at the beginning is probably the most plausible one.
Yeah. So then the other question that I have is, and you may not be able to answer this because of affiliations or other things. So if you need to dodge the question, it is perfectly acceptable. Is there anything like this that's on the radar out there now, like the Human Genome Project, that's sort of in that state that it was, let's say, in the 80s, that, somebody's trying to make a go, no-go decision on some basic science that, 30 years from now, if we were doing this, we would be talking about how that would be different. We're just on the front end of the fork instead of on the back end of the fork.
There are a couple of initiatives that bear some similarities. I think some of the background politics are maybe not quite as difficult, because I think in large part a lot of the utility of this is understood. But there is interest in understanding more about human gene expression. That's one of the projects that's really being worked on. There's not necessarily a Human Genome Project analog for that necessarily, but this is definitely an area of active inquiry. So, kind of when we started out, if you look at some of the source material in the 80s and 90s, when we're talking about the Human Genome Project, the estimates of the number of human genes were generally somewhere between about 50,000 genes and about 100,000 genes. Because it was thought that, well, we're humans, obviously we have way more genes than all these other mammals and other organisms that we consider to be, quote unquote, lower than us. As it turns out, humans have about 22,000 genes, which kind of makes us nothing special in that respect. And but we are different, no question there. But I think that there was a little bit of biological hubris there when we assumed that we must have more complexity of our genetics. And there probably is a little more complexity, but it's different complexity. And so one of that is the way human genes are expressed, how genes are turned on and off, and how genes are expressed in different organ systems, different tissues. And that's an area that we still don't completely understand. And we know that that's an area of a lot of importance. The other and some of that is understanding what happens to the protein products that are made. Some of that happens with what happens to the RNA products that are made from DNA, kind of the intermediary. And so that's an area of interest. The other thing that's of interest that is a little more like the Human Genome Project is the microbiome, is looking at the human microbiome. And what is the microbiome? And we could do a whole other hour on this, Don. I don't know if the listeners really want to hear this, but this is potentially a whole different discussion. And so we have microbes all over us. We have microbes on our skin. We have microbes in our gastrointestinal tracts. We have microbes in other parts of the body. And these microbes are actually they're very important. It's very important that we have certain species we know are very, very protective. Certain species are things that we were used to seeing, and certain species that if they get in overabundance can cause problems. And so we are gaining some understanding of what normal looks like there, or what optimal looks like there. But we also know that our microbiome has changed over the last 100 years. A microbiome of a person's GI tract in 1920 versus 2020 looks a lot different. And there's probably some negative effects from that. But we don't necessarily know what those are completely.
Right.
And so understanding those species and understanding the genetics of those species is really important too.
Yeah. And to me, that's that thing I can't get out of my head. I don't know if that's a good or a bad thing. Is just, again, the application versus the basic here. That you're talking there
Absolutely.
about more of the application. And the application, of course, is where we actually see the benefits. And I appreciate you sharing that statistic about the ROI of $1 to like $170. In most instances, if all of us knew we just had to put that dollar coin over in the slot machine in the corner, it was going to return 170 every time, we'd be going around looking for dollar coins, because we would want to stick as many of it as we could there. The other thing I just wanted to ask, I guess, just briefly about, that sort of goes along those same lines, but at the same time is sort of different, is we talk about mapping the human genome. And so one of the things that I have in my head is that part of the technology that you described there, it's a little bit like what happened as we moved from physical maps to like GPS in terms of what you're able to do in terms of how it works. Is that a fair analogy?
Yeah, you can draw that analogy. I think that's a fair way to sort of understand the difference in complexity, especially as we move from kind of those rough maps of the human genome with just those markers that were used for linkage analysis. I think that's a very rough map. Whereas I think the Human Genome Project, where we're actually sequencing those regions, and again in entirety, and I'm going to use entirety in quotes. We're sequencing those. It's just it's a higher resolution map, and so you don't have to pull your paper map. And I still have my paper map of the state of Kansas in my glove box. I don't think I've pulled that thing out in 10 years.
Yeah, well, and I know
Yes.
even GPSs. I remember the first GPS that I had that mounted in a vehicle. The time that sat on the dash, or
Oh yeah.
up to the windshield or whatever. And I was in sales at the time, so it was really cool to have that. That made getting versus what we have this thing in the Houston area called a key map, which is the city map. You may have something similar in Kansas City. That I became I could do some key map kung fu kind of thing. In terms of getting around and being able to navigate that pretty well. The GPS totally replaced that. Well, now we don't even use a separate device in most instances, or it's built into our car, or we just use it out of our phone, whatever the case might be. That's changed a lot. Part of the reason I asked that question, Eric, is that those listeners that stick to the end sometimes find out why we name episodes the way that we are. What I'm tempted to name this episode is Recalculating, like you hear from a GPS.
Oh, yeah.
If
like
if we had not gone down the Human Genome Project, we sort of would have been doing recalculating that we don't have to do now, because we have the base map that allows us to understand some important things that we wouldn't have had otherwise. So that's the other reason for asking my question, is just to see if the analogy that I want to use in the title makes any sense in terms of the actual things that we've discussed.
Absolutely, and I think we can always we can look at some of the original documents for the Human Genome Project with modern eyes and say, well, we were probably a little naive going into this, but I think that's okay. I mean, I think it's okay that you're a little naive going into it, and kind of making some of those assumptions of what we're likely to find and not find. Because I think if you aren't a little naive going into this, we're probably not pushing the envelope enough.
Right.
We're probably not being as adventurous with our technology and kind of pushing things forward. And we always kind of know, anytime we go embark upon a big sort of moonshot program, once we kind of get that project going and kind of understand the ramifications of that, those first couple of years are always going to look a little naive. And I think that's okay. But I think, it really was a visionary project. And in my opinion, it's paid off. Not necessarily in the same way that we thought it was going to pay off, but it certainly paid off.
Yeah, again, using the moonshot analogy, which always works for me, because I'm of that generation. Started before I was born, but the first moon landing is about a year and a half after I'm born, so I'm in that and have an affinity for that. It gave us all types of, fibers and technologies and computer technology. It gave us Tang. So you're going to get all kinds of things, out of a space program, and some of them are going to be more valuable than others, right? Eric, I really have enjoyed having you on the podcast again today. Always want to have you back. So anytime you have a topic suggestion,
it's a pleasure, Don. Happy to come back.
Please know we look forward to that. And as I was mentioning to Eric off here, we receive a lot of nice words about episodes, but personally, people I know have walked up to me and said, make sure you have that guy on again. I really, really, really enjoyed that episode. So I was looking forward to, not that I don't look forward to doing the other episodes, but I was really looking forward to doing this one. So I'm going to close this out here. Thank Eric again. And Eric's wearing his Kansas City Royals shirt as I'm looking here. He's from Kansas City. I'm wearing old school Astros Colt 45s hat. So we're into baseball season. So he and I may talk a little bit off the podcast about some baseball stuff, but we won't bore you with that. Once again, we thank you for joining us here on A Fork in Time. I really appreciate of you as a listening audience. Again, if you want to learn more about the podcast, visit the website. That's www.aforkintimepodcast.com. As Lex and I always say, the A is important, the podcast is important, or else you won't get to the right place. We have done some revamping there. There's nothing fancy or nothing special about the website, but if you haven't visited in the last week, you'll go there. You'll see that it has a different look and feel. And I'll just mention, it also has some things on there now about the new exercise that we're looking at starting here in June, which is the new podcast, the Room Where It Happened. And so you can learn a little bit more about that. So I wanted to get that mentioned in here. Once again, Eric, thanks for your time today. Thanks for joining us. And we're going to wish our listeners a good day and hope that they join us next time.
All right. Thanks, Don.
Thanks.
Thanks for listening to A Fork in Time, the alternate history podcast. Learn more and provide feedback by visiting our website at www.aforkintimepodcast.com. Connect to us on Facebook at facebook.com/aforkintime or follow us on Twitter at AFIT Podcast. If you want to support the show financially, visit our Patreon page at patreon.com/aforkintime. We hope you will join us next time.